So while our team finishes collecting your ballots, please allow me to introduce the next speaker. This is, at a young age, she has already always been fascinated with science. She started out as an amateur botanist and ornithologist, until she read a book about memory research as a freshman in college and never turned back. Today, she's the first female director of the Center for Neuroscience and a professor of neurology and of neurobiology, physiology, and behavior. She's the co-champion of the UC Davis Healthy Brain Aging Initiative and a principal investigator of the UC Davis Conte Center, funded by the National Institute of Mental Health and one of only 15 such centers in the country. Her research examines how circuits in the developing and aging brain are formed and sculpted. Her lab is one of the few in the world that studies the role of immune molecules in regulating synapse formation and function, as well as the molecular pathways that underlie risk and the resilience of offspring with brain disorders following maternal immune activation. She's a trailblazer not only in her own research, but also through her mentorship and training of young researchers, advocating for diversity, equity, and inclusion in science. Please join me for in welcoming Dr. Kimberley McAllister.
Okay. Can you hear me better? That's better, okay. If it is too loud or difficult to hear, could somebody in the back raise their hand and we'll figure that out? There's all sorts of feedback that we're getting from a bunch of these different sound systems, so I really apologize for that. Okay, so I am going to start off my talk with a very simple question. At least a simple sounding question. What is memory? As I get older, when I think about this question, I actually think more about what is forgetting. And I think about all the things that I forget all the time, increasingly as I get older, right? Where's my iPhone? How many times have you said that? I say it all the time. Where's that darn TV remote? Where did I put that? I don't remember. And the biggest one for me now, where did I park my car when I come out of a store, right? So as annoying and as common as these examples are, they really don't illustrate the real importance of memory. The ability to learn and remember is fundamental to every single act of daily living. Our memory enables us to learn from the past, to stay oriented in the present, and to plan for the future. So as Stephen King put it, a person's memory is everything, really. Memory is identity. It's you. So who am I? I am a daughter, I'm a sister, I'm a mother, and as you've heard, I'm the director of the Center for Neuroscience. Without my memories, I would not be me. I would be me, but I wouldn't have my identity. And that's honestly the harsh reality of what people who suffer from Alzheimer's disease and dementias face. So this picture, which is similar to the picture that Lucas used in that beautiful talk a few minutes ago, really poignantly illustrates the loss of identity that accompanies Alzheimer's disease. And as Lucas pointed out, this terrible disease is way too common, and it's way too devastating. Alzheimer's disease is the 6th leading cause of death in our country, and it's the only top 10 cause of death that cannot currently be prevented, treated, or cured. As Lucas mentioned, it's affecting about 5 million people right now in our country, but this rate is only going to increase. It's going to increase, they estimate, to 7 million by 2030, and a whopping nearly 14 million by 2050. So we clearly need to develop new treatments as fast as we possibly can to stem the suffering that we know is coming in a tsunami of disease by 2050. So how can we develop approaches to improve memory and to prevent diseases like Alzheimer's disease? Well, there are a number of simple steps that we must take in order to achieve this goal. And a lot of it involves looking into the brain to identify where memory actually occurs in the brain. Second, we really want to determine then how memories are established within circuits within those brain regions. Third, we want to identify how memory loss actually occurs in disease. And then, fourth, most important, we want to develop strategies to treat and prevent memory loss in disease. So the strategies that we think we will be able to identify in step 4 really rely on the other steps. They rely on allowing us to come up with ways to fix the things that go wrong in disease that we'll identify in step 3, using approaches that are identified in step 2 to restore the typical way that memories are made in the brain and within the very specific brain regions where memory actually occurs. So where does memory occur in the brain? About 30 years ago, we did know a little bit about where memory occurs. And when the center was launched 30 years ago, our basic knowledge about where it occurs came from historical studies from psychologists who had studied patients that had lesions in the brain. And they had lesions based on either injury or on surgical resection for treating diseases like epilepsy. And some of the most important discoveries were made by an inspiring scientist, a psychologist named Dr. Brenda Milner, who's shown here on the slide. And she studied one particular patient who had a really fascinating type of memory loss. Some of you may have heard of this patient. He went by his initials, we refer to him by HM to protect his privacy. Now this man had intractable epilepsy, and to treat that epilepsy at the time, what they did was to resect a region of the brain that I'm circling here on the slide, right around what we now know as the hippocampus. And so that area was completely missing in HM. The surgery improved his epilepsy, but it led to a major problem with memory that was really interesting. HM was no longer, after surgery, able to make memories for new events. But he could remember events from his past that he had had before the surgery. And so what Dr. Milner concluded from this was that memories for new events must occur in the region of brain that was taken away in the hippocampus, but memories for past events must somehow not involve the hippocampus anymore and instead be stored in this area of the brain outside of the hippocampus. Okay, so since HM, our field has developed really great techniques that allow us to look into the brain in healthy individuals to see where memory occurs. And so functional magnetic resonance imaging, or fMRI, was developed actually only about 30 years ago. We all just assume it's been around forever, but it was only 30 years ago that people started using this. And it's one of the most widely used approaches to start to study where memory occurs in the brain. Now, we have several faculty members here, some in the audience, who use this approach and who are leading world experts in human memory. And at the Center for Neuroscience, one of our experts is pictured here, Dr. Charan Ranganath. So fMRI has begun to reveal the activity patterns that are involved in creating and recalling a specific memory. As memory is encoded and initially retrieved, the hippocampus lights up, which is this area that I'm pointing at right here with the pointer. But long-term memory storage occurs across many different brain regions depending on the memory. Different types of memory involve different sets of areas. So for example, long-term memories for words might be stored in the front of the brain, whereas long-term memories for scenes might be stored in the back of the brain here. Now, Dr. Ranganath's lab, he asked me for a favor, and they actually need some help with soliciting volunteers. So his lab is interested in understanding where these different types of memory are encoded, but he really wants to understand how that encoding and that long-term storage changes over healthy brain aging. And so we know a lot about what happens at the end of life, we know a lot about what happens early in life, but we don't know much about what happens over the process of healthy aging. And so he has a study that he needs to recruit volunteers for. So if you are over age 60 and you're interested in being part of a neuroscience study, please let me know. We've got flyers down here, and Dr. Ranganath's lab would be really happy to have you volunteer if you're interested. Okay, so once we start to have a sense for where memory might be encoded, we want to ask the question of where does memory actually occur within those brain regions. Now, the search for the physical manifestation of a memory in the brain, which is called the engram, has lasted for more than 100 years since the engram, that term, was first defined. And it was initially proposed that memories are stored in subsets of cells within specific brain regions that are connected to each other. And so in order to be able to find those cells, we have to be able to resolve them. But fMRI doesn't have the resolution where we can actually see single cells. And so while fMRI told us where to look, generally within the brain, we had to turn to animal models to start to figure out how memories were actually encoded within those regions. And so this search for the engram has been driven by technological developments, similar to a lot of the technological developments that you've all heard about over the course of the day today. So in terms of this particular search, it involved mouse research. And before 1980, it involved an approach that was really similar to what psychologists had used in studying HM. Basically, scientists removed large parts of the brain and looked to see whether memory was still there or whether it was gone. And over time, those techniques were refined in the 70s and 80s, where they could create targeted lesions of more specific brain regions, narrowing where the memory might be stored. And then in the 1990s, scientists developed ways to silence neurons within a specific brain region, again, now getting closer to identifying the neurons that are actually involved in creating that memory. But it wasn't until 2009 that techniques were developed that really allowed us to start to identify the actual neurons that are part of an engram, of the physical manifestation of a memory. And one of the key advances that allowed us to do that was the development of methods in mice that allowed us to identify the neurons that were active at the time of learning, and then to express genes within just those individual neurons. Okay, so what you're looking at here is a picture, a slice through the hippocampus. This is packed full of cells. You don't see most of them, but you see a subset of these little green neurons. They're really beautiful, right? These are neurons that are expressing a green fluorescent protein that was turned on only in the neurons that were active at the time of learning a task in a mouse. So what's great about this technique is that it doesn't just allow us to turn on markers like green fluorescent protein, but it also allows us to turn on genes that we can use to control the activity of the cells, just like Dr. Kim described in her talk, a beautiful talk earlier. And we have a faculty member that's actually in the room who is very famous, Dr. Brian Wilchin, Brian Wave, and I'm embarrassing him, but he's a member of the Center for Neuroscience, and Brian was instrumental in developing and using this technology to start to identify the engram. And what Dr. Wilchin and his colleagues can now do is pretty phenomenal given where we were in studying memory 30 years ago. What they can do now is actually to go in and not only identify the neurons that are involved in a specific memory, but they can erase memories by silencing those specific engram neurons, and they can induce artificial memories by stimulating those engram neurons. So whether we're going to be able to do this in humans is an open question. And as Dr. Kim mentioned, this is something that people are starting to think about how to adapt these approaches to be able to be used in humans. But the idea eventually would be that in early Alzheimer's disease, as individuals start to lose their memories, we might be able to use these approaches to reawaken memories in those patients. But regardless of whether this will turn into a treatment, it has told us a lot about how memory, where memory occurs, and it's starting to give us some insight into how. So the way that the engram works is that it's made up of connections between a small subset of cells within a number of different brain regions. These are initially hooked up to the hippocampus, and as memories are made, they fire together, they are active at the same time, and that strengthens those connections. Over time, during recall, as the memory is recalled over and over again, that then leads to strengthening of the connections in the areas of the brain that are important for storage, and the removal of the connections to the hippocampus. And that really begs a really interesting question. How can the strength of some connections be modified during memory formation and storage? Other connections from cells that are neighboring not be affected at all, and how can it lead to the removal of other connections? So in the 1950s, one of the most influential ideas for how this might work was proposed, again by another psychologist, Donald Hebb. And Donald Hebb proposed that neurons that fire together wire together. And so what that means is that cells that are active during the formation of a memory will have their connections strengthened so that they remain connected to facilitate the recall of that memory. Now, it wasn't until about 30 years ago that we started to gain any understanding of how synaptic strengthening and weakening might actually occur. And the process that underlies the long-term modification of the strength of synapses is called synaptic plasticity. And there are lots of forms of synaptic plasticity, but two in particular, long-term potentiation strengthens those connections and long-term depression weakens them. And so the synapse should be familiar to you now after the talks earlier. Basically, the way this works is that information is transferred across cells by the release of neurotransmitter that then binds to receptors on the recipient cell and therefore transmits information. When these connections are stimulated rapidly, like would be happening in a memory, you end up with an increase in synaptic strength or potentiation of your connection, which means that basically it's releasing more neurotransmitter and there are more receptors to bind to, so the signal is just a lot louder. So it's really actually astounding how much progress has occurred in our understanding of the cellular and molecular mechanisms that mediate this. This is just the tip of the iceberg here. In fact, there have been 17,000 papers that have been published since 1992 just on the mechanisms of long-term potentiation alone. It's a lot of papers. It's a lot of work. So several faculty at UC Davis have contributed to this knowledge, including myself and Dr. John Gray, who studies some of the most important neurotransmitter receptors that are involved in LTP called the NMDA receptors. So one of the most exciting advancements in the field has been the discovery that strengthening connections by LTP during memory formation can also cause more connections to form, which further solidifies that engram. So what you see here is a slice through the hippocampus. You see a number of these green cells. That's just a subset of the cells that are now labeled with this green fluorescent protein. And if you come and look at just this little tiny box here, at higher magnification, you can see that process of the cell, the dendrite, actually has a lot of little protrusions on it. These are called dendritic spines, and these spines are where synapses form onto these neurons. So about 30 years ago, again, scientists developed really sophisticated approaches to allow us to image these structures in living tissue. And another one of the center's faculty, Dr. Karen Zito, developed a way to look to see how these spines change with long-term potentiation. And what she found is that when you stimulate a spine, you can actually, if you stimulate it with a protocol that would be found like during memories or forms, long-term potentiation, you end up with the spine getting much larger, and it stays larger over a long period of time. So ultimately, though, to restore memories, we're going to want to increase the strength of synapses. And to do that, we need to know, as Dr. Kim really beautifully pointed out, we need to know what the components of those synapses are and how those components are modified by synaptic plasticity. So while we generally knew how synapses were potentiated about 30 years ago, we didn't have any idea about how complex these synapses actually were. And we've gained a lot of really surprising understanding of that over the last 30 years. So don't get scared. This is a picture. I'm not going to go through it. But this is a picture of just the tip of the iceberg as far as the complexity of molecules at the synapse. It turns out that there are over 1,000 different kinds of proteins that are found at synaptic connections in, at least just for excitatory synapses in the brain. And there are many different kinds of connections. And so while this looks a little bit overwhelming, and even for those of us in the field, this is pretty overwhelming, the sheer complexity of this is incredibly beautiful to those of us that want to come up with mechanisms to improve memory. And that's because each one of these molecules actually has the potential, if we understand it, to be able to be used as a target for developing new drugs against that can then modify synaptic plasticity. Okay, so how can this information be used to develop new treatments to improve memory and prevent diseases like Alzheimer's disease? Well, of course, first, we need to understand what's wrong with the memory machinery in this disease, and then we need to fix it, right? So for Alzheimer's disease, it's been known for more than 30 years that there is severe degeneration of the brain and loss of synapses in the brains of these patients. But it wasn't until about 40 years ago that some of the causal pathology was actually identified, what actually leads to this loss of brain tissue that you can find in severe Alzheimer's disease. So some of you know about this. You've heard about the plaques and tangles. So it was found in the 1980s that these are comprised of amyloid and tau protein. And it wasn't until about 30 years ago, kind of remarkably, that the first genetic risk factors were identified. I know this because I was at Duke University starting my graduate career when they identified some of the ApoE genes. Also around this time was the very first time that the different stages of changes in the brain during Alzheimer's disease was identified. It didn't even happen until about 30 years ago. And it wasn't until 25 years ago that the first mouse model of a risk factor for Alzheimer's disease was actually established. So this field of understanding what's going wrong in disease has not been around for that long, but it's picking up pace. And so only recently, in the last few years, have we really started to gain insight into what might be going wrong at these synapses so that we can start to figure out how to fix it. And so this is a rather complicated figure that I'm going to show, I'll take you through. It illustrates the results from a study that I find really exciting. So this is a neuronal cell body. This is a normal synapse, so this is where you make connections and you have neurotransmitter release. In the Alzheimer's brain, in an early brain, what you see is you do see the accumulation of amyloid and amyloid plaques. And for many years, it was thought that it was these plaques that were actually causing the degeneration and the disease. But it turns out that it's actually in early disease, it's these little A beta monomers that get secreted from the neurons and that actually go in and they come into the synapse, they bind to molecules at the synapse, and they cause those synapses to be hyperexcitable. Hyperexcitability might sound good sometimes if you want some energy, but it's not good for neurons because neurons die when they're hyperexcitable. And so this is just one glimpse of a new discovery just over the last 3 years of how some of the basic pathology in Alzheimer's might be altering synapses to cause synapse loss early on. But as Lucas told us in the data blitz, it's not just amyloid that leads to Alzheimer's disease. It's also inflammation in the brain. And so about 20 years ago, it was recognized that inflammation plays a role in Alzheimer's disease. But it wasn't until about 10 years ago, only 10 years ago, that the cell type, the immune cell type that plays this role was identified. And these are called microglia. But we know very little about how inflammation causes synapse loss and leads to loss of memories in Alzheimer's disease. So the lack of understanding of how synapses are lost in Alzheimer's has led to a lack of progress in developing new treatments. So current treatments that many of you may be familiar with for mild to moderate Alzheimer's disease are basically anticholinergics like Aricept. To moderate severe Alzheimer's disease are drugs like memantine. But that's kind of it. How can that be? There are a number of current clinical trials that are now focused on developing ways to alleviate the pathology from amyloid. So people are using antibodies to A beta. There's a recently FDA-approved drug for doing just that is quite controversial in the field. But I think we could all agree that new treatments are definitely needed. New targets are needed. And so a number of us are really interested in synaptic proteins that mediate plasticity and memory, and that are altered by A beta and inflammation, as looking at those as new targets for developing new drugs to treat Alzheimer's disease. And so I've got one more data slide, and then I'm going to wrap up. And this data slide basically tells you about why, one of the reasons that I care about this from a scientific perspective, because our lab has just recently moved into this field. And so we are interested not in the immune cells that are involved in synapse loss, but we're interested in immune molecules that are found at synapses and how they can lead to synapse loss. And the molecules that we focus on are major histocompatibility complex 1 molecules. So these are molecules that some of you may have heard of. If you read about immunology, they're found on all of the cells in your body where they help you to fight off infection. A number of years ago, my lab showed that MHC molecules are actually found at the synapse where they regulate synaptic plasticity and lead to synapse loss. So a talented postdoc in my lab here named Gabby Sell has recently discovered that MHC molecules are actually upregulated by amyloid beta in Alzheimer's disease models. And that upregulation leads to a decrease in plasticity molecules at the synapse and causes synapse loss and memory deficits. Now, this is exciting because she's now proposed an intervention where she can prevent that increase in MHC, which then might rescue these effects. And she has some recent data that's very exciting, suggesting indeed that's exactly what happens. So if we prevent this increase in MHC, we end up with rescuing the plasticity molecules, they come back to normal levels, and we prevent synapse elimination and memory deficits. Now, we're very far away from looking at this as a treatment, but this is really exciting because once we start to find molecules like MHC that might be able to rescue these deficits, we can then start to spend a lot of intense effort trying to figure out if these are good targets for developing new drugs against. And there are many labs, this is just one example that we're excited about, but there are many labs across the world, hundreds, that are identifying these targets and are following them through to see if we can develop new treatments. So it's very exciting. Okay, so I hope that you can see how far we've come in our understanding of each of the different steps that we need to follow to come up with these new treatments over the last 30 years, and that the pace of discovery really is only accelerating. We've gone from knowing very little about where and how memory occurs in the brain and even what goes wrong in Alzheimer's disease, to being on the cusp of developing a wide range of strategies to treat and prevent memory loss during disease. Those strategies will include things that you've heard about over a number of talks this afternoon, including brain stimulation, altering activity within the specific engrams maybe in the future, and developing whole new classes of therapeutics. So the outlook for the next 30 years is finally, I can say, with real enthusiasm and scientific rigor, it really is that we finally have real hope for the millions of people that are going to be afflicted with Alzheimer's disease in the next 20 years. Thank you.
Hi, thank you so much for the presentation. So a quick question on the controversial new drug, the Aduhelm, I believe it's pronounced. So if the main purpose is to try to reduce the amyloid beta plaque proteins, how come so many patients are developing more neurological issues or even dying? What causes that, and why can't that be circumvented?
Right, so these are the questions that you exactly want to ask when you're doing clinical trials, right? You think that you have a candidate, you test it, and then sometimes you get effects that you're not expecting, which is why we do clinical trials. I'm not an expert in that exact drug or privy to all of the data that underlies that, so I can't answer your question specifically. But I can say that when you alter anything in the brain, you're going to have unintended effects, right? Especially if you're not targeting it to the specific cells that that are at the root of the memory loss, okay? So what Dr. Kim talked about is a much more targeted strategy where you wouldn't be affecting all of the cells in the brain, really in the body if you take a drug, but instead it would be targeted directly at the cells that might actually be the source of the issue. Right now, and for the next short foreseeable future at least, we're going to have to deal with this and find and go through a lot of different candidates and hopefully find ones that have less side effects and better outcomes. Yeah, but there's a lot of potential and a lot of things that will be coming down the pike in the future. Yeah. Yeah, one thing that I would also say about that is that I was really surprised when I looked back over the last 30 years, because I thought that we would be, I've been frustrated as to why we aren't further along in having new treatments. But when you look at what we knew 30 years ago and what we needed to know to be able to come up with new treatments, this field is moving fast. We need to move faster, we all know that. We feel that urgency, but we're really, really on the cusp of making big discoveries. And for those graduate students in this room right now, you're going to be the ones that do that.